Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. Today, we're going to talk about a very important part of the MCAT for both the ChemPhys and biochem sections. Biotech and protein analysis are very important topics to understand for the MCAT. Scientists often isolate proteins from their natural sources to study their structure, function, and properties in detail. Protein isolation is a critical step in biochemistry research, and several methods are used to separate and purify proteins from complex mixtures. In this video, we'll explore 3 common methods for protein isolation: electrophoresis, polyacrylamide gel electrophoresis, and isoelectric focusing, IEF. Let's start with electrophoresis. This is a widely used technique for separating and analyzing proteins based on their charge and size. It involves applying an electric field to a sample containing proteins, which causes the charged molecules to migrate towards the oppositely charged electrode. The rate of migration depends on the size and charge of the protein, allowing for separation of proteins based on their physical properties. There are 2 types of electrophoresis: native and denaturing. Native electrophoresis is performed under conditions that maintain protein structure, while denaturing electrophoresis involves disrupting the protein structures using chemical reagents like SDS, sodium dodecyl sulfate. SDS coats the protein in an overall negative charge. This will cause the protein to migrate towards the anode of the gel in the electrophoresis machine. Negative ions always move towards the anode, and cations always move towards the cathode, no matter what. The larger proteins will move more slowly and therefore will be located closer to the top of the gel, whereas smaller proteins can more quickly zip down towards the bottom. So we're going to find smaller proteins at the bottom of the gel and larger proteins near the top of the gel. Next, we have the Page gel. Page stands for polyacrylamide gel electrophoresis. So gel electrophoresis is the exact same as electrophoresis. The last part we've already explained. So really, we're just talking about the PA right now. Page is a type of electrophoresis that uses a polyacrylamide gel matrix to separate proteins based on their size. First, the sample is loaded into a gel that is poured into a rectangular chamber and then subject to an electric field. The proteins migrate through the gel matrix, with smaller proteins moving more quickly than the larger ones. Once the proteins have migrated to the desired distance, the gel is stained to visualize the separated protein. Page is widely used in biochemistry research to separate, quantify, and analyze proteins. There are 2 types of SDS Page: denaturing and native SDS Page. Denaturing SDS Page will use a reagent like beta-mercaptan ethanol to reduce any disulfide bonds between cysteine to ensure tertiary and quaternary structure are destroyed. However, sometimes a native SDS Page is preferred, so we can see the size of the entire protein while still having a coded negative charge. To make things more complicated, we can have a native page where no SDS is added. This will cause the protein to migrate based on its charge and size. To make things more complicated, we can also have a native page where no SDS is added. This will cause the protein to migrate based on its charge and size alone. Finally, we have isoelectric focusing, or IEF. IEF is a type of electrophoresis that separates proteins based on their isoelectric point, PI. Check out our earlier video on this. The PI is the pH at which the protein has no net charge, and thus it will not migrate in an electric field. In IEF, a pH gradient is established in the gel matrix, which separates the proteins based on their isoelectric point. Once the proteins reach their respective PI points, they will no longer migrate and will become immobilized in the gel matrix, shown by these bands here. IEF is a highly sensitive and can separate proteins with high resolution based on their isoelectric point. This makes it very useful for studying protein structure and function. In summary, protein isolation is a crucial step in biochemistry research, and several methods are used to separate and purify proteins from complex mixtures. Electrophoresis, Page, and IEF are 3 common methods of protein isolation that rely on the physical properties of proteins, such as charge and size, to separate them from other molecules. These methods allow researchers to analyze and study proteins in detail, providing insight into structure, function, and properties. The reason you care is that all of these researchers are what are making the passages for biochem and ChemPhys. So by understanding what these passages are trying to say, you'll be better able to interpret them and get more questions right on the MCAT. Thank you so much for watching our video on these common biochemical techniques, and I will see you next time.